Hello friends, today we'll learn about Bordetella. We'll study in this video about the introduction, morphology, culture, biochemical reactions, resistance, antigenic structure, variation, pathogenesis, epidemiology, laboratory diagnosis, treatment, prophylaxis, and Bordetella parapartosis, Bordetella bronchioseptica. Next, we'll study about Bordetella avium. So, let's begin with introduction. The genus Bordetella contains very small gram-negative cocobacilli which were formerly included in the genus Haemophilus. As they grow, they do not as they do not require X and V factors for growth and are antigenically different from Haemophilus, they are separated into another genus Bordetella. Bordet and Gengo in 1906 isolated the organism Bordetella pertussis from children suffering from whooping cough. Now let's study about the species. B. pertussis causes whooping cough. B. parapertussis responsible for milder form of whooping cough. B. bronchiceptica responsible for both for about 0.1% cases of whooping cough of milder type. B. avium causes respiratory disease in turkeys. Now let's study about Bordetella pertussis. Let's study about the morphology. B. pertussis is a small ovoid 1 to 1.5 micrometer into 0.3 micrometer gram negative cocobacillus. It is non-motile and non-sporing. It is capsulated but tends to lose the capsule on repeated subcultures. Bipolar metachromatic granules may observe on staining with toluidine blue. Now let's study about culture. It is aerobic and cannot grow anaerobically. Optimum temperature for growth is 35 to 36 degrees centigrade. The organism requires a complex medium for its primary isolation. Bordet gengo glycerol potato blood agar is a commonly used medium. After incubation for 48 to 72 hours, colonies on this medium are small, smooth, opaque grayish white, refractile, resembling bisected pearls or mercury drops. A hazy zone of hemolysis surrounds the colonies. Blood in the medium is apparently not to provide additional nutritive factors but neutralizes inhibitory substances like toxic fatty acids. Charcoal or starch or both may also be incorporated to neutralize these toxic constituents. Charcoal blood agar medium is an alternative medium used for primary isolation of this organism. It does not grow on nutrient agar. Now let's study about the biochemical reactions. B. pertussis is biochemically inactive. It does not ferment sugars, form indole, reduce nitrates, split urea or utilize citrate. It is oxidase positive and usually produces catalase. Now let's study about the resistance. It is killed by heating at 55 degrees centigrade for 30 minutes, drying and disinfectants. It survives outside the body in droplets for few hours. It retains visibility at low temperature, that is 0 to 4 degrees centigrade. Now let's study about antigenic structure. Agglutinogens. Freshly isolated strains of B. pertussis possess heat-labile antigens associated with capsule K antigens designated 1 to 14 factors or agglutinogens. All strains of B. pertussis carry factor 1 and 1 or more of the other factors. Factor 1 to 6 are present only in strains of B. pertussis. Factor 7 is found in all strains of three species of Bordetella. Factor 12 is specific for B. bronchiceptica and factor 14 for B. parapertussis. These antigens are useful in serotyping of strains for epidemiology purposes. Some of the agglutinogens have been recognized on the fimbriae. B. pertussis have 8 agglutinogens, 1 to 6, 7 and 13. These type specific antigens are under the control of virgin. Agglutinogens promote virulence by helping organisms to attach to respiratory epithelial cells. As strains causing infections are the type 1, 2, 3, and 3, it is essential that pertussis vaccine strains should have factor 1, 2, 3. Now let's study about lipopolysaccharide. It is a heat stable lipopolysaccharide endotoxin of the cell wall. It is not protective. It is common to all smooth stains of all the three species of Bordetella. Now let's study about the heat labile toxin. Heat labile inactivated at 56 degrees centigrade into 15 minutes. Toxin is present in all Bordetella. Pathogenic role of this toxin is doubtful. It is cytoplasmic protein. Tetrachial tracheal cytotoxin. Let's know about the tracheal cytotoxin. It is derived from the peptidoglycan of cell wall and is present in all Bordetella. It causes damage to respiratory epithelial cells and therefore makes a person more prone to secondary infection. Now let's study about pertussis toxin. It is a heat labile exotoxin called the pertussis toxin. It is present only in B. pertussis. It is a major virulence factor. It is also known as lymphocytosis producing factor, histamine sensitizing factor, and isolates activating protein. It has a wide spectrum of biological activity. It is apparently responsible for many of the signs and symptoms of pertussis. It also causes profound lymphocytosis in pertussis patients. PT has a molecular weight of PT means pertussis toxin has a molecular weight of 170,000 and is composed of 6 polypeptide chains, that is S1 to S5 with 2 copies of S4. PT has got 2 units A and B. The A unit being the enzymatically active moiety and B the binding component. The A unit is made up of S1 while B unit consists of the remaining 5 polypeptide chains. Pertussis toxin can be toxoided. PT toxoid is the major component of acellular pertussis bacillus. Now let's study about adenylate cyclase. AC. Only one of adenylate cyclases has the ability to enter target cells and act as a toxin. This is known as AC toxin. It acts by catalyzing the production of CAMP by various cells. It is produced by all the three mammalian Bordetella. Now let's study about filamentous hemagglutinin, in short FHA. It is present on the bicellary surface. It mediates the attachment of the bacterium to the ciliated epithelial cells of the respiratory tract. It also adheres to erythrocytes. Antibodies directed against FHA are protective. FHA is used in acellular pertussis vaccines along with PT toxoid. Now let's study about paractin. It is an outer membrane protein present in virulent strains of B. pertussis. It included in acellular pertussis vaccines. Antibody to paractin can be detected in blood after infection or immunization. Now let's study about variation. Freshly isolated strains are the smooth form, that means in phase 1. On subculture, they undergo loss of surface antigens and become phase 4, which is the raw avirulent form. They have to pass through phase 2 and phase 3 before becoming phase 4. A reversible change in the capsular antigen has been described. The organism may occur in one of three modes X, I, and C. Each mode has a characteristic surface antigen. These modes refer to the color of the colonies on the Baudet Gengo medium X for xanthic yellow, or C for cyanic blue, and I for intermediate. On the Bordet Gango medium, press isolated always occur in X mode. Now let's study about pathogenesis. B. pertussis infection is predominantly a childhood disease and is known as whooping cough. 95% cases of whooping cough are due to B. pertussis, 5% by B. pertussis, and only in about 0.1% cases B. bronchoseptica is responsible. In human beings, the source of infection is the patient in early stage. The incubation period is about 1 to 2 weeks. Infection is transmitted by droplets. The disease usually lasts for 6 to 8 weeks. It consists of three stages, namely the catarrhal, paroxysmal, and convalescent, each lasting approximately for 2 weeks. Although cases are maximally infective during the catarrhal stage, but clinical diagnosis in this stage is difficult. During the paroxysmal stage, the virulent spasms of continuous coughing is followed by a long inrush of air into the almost empty lungs with a characteristic whoop. During convalescent stage, the frequency and severity of coughing gradually decreases. Now let's study about complications. The disease is self-limiting. Complications may be maybe first point is subconjunctival hemorrhage due to the pressure effects of violet coughing. Next is bronchopneumonia and lung collapse. Third point is convulsions, coma. Convulsions and coma. Now let's study about epidemiology. Whooping cough is predominantly a pediatric disease. The incidence and mortality are the highest in the first year of life. The source of infection is the patient most infective during early stage. The mode of infection is by droplets. Whooping cough is one of the most infectious of bacterial disease. Healthy carriers have not been identified. Although natural infection confers immunity, but second attack may occur. Reinfection in adults may be of severe form. Now let's study about the laboratory diagnosis. First is microscopy. Microscopic diagnosis is made by demonstration of bacilli in respiratory secretions by the fluorescent antibody technique. Now let's study about the culture. In the early stage, bacilli are most abundant in the upper respiratory tract. They are scanty in the paroxysmal stage and are not demonstrable during convalescence. For culture some methods have been used for collection of specimens. Let's know about the methods. First is parnasal swab. A swab is passed along the floor of the nasal cavity and material collected from the pharyngeal wall. The swab is inoculated on Bordet gango medium or charcoal blood agar. Nasopharyngeal aspartate collected through a catheter attached to a syringe in a better alternative. Now let's study about the cough plate method. A boarded gango culture plate is held 10 to 15 cm in front of patient's mouth during a bout of coughing so that the cough droplets directly inoculate the culture medium. Now let's study about postnasal swab, or you can say peroral swab. A west postnasal swab is passed through mouth to collect posterior pharyngeal wall secretions. Precautions should be taken to avoid salivary contamination. Fatty acids present in cotton may inhibit the growth of the bacilli, and so it is better to use Dacron or calcium alginate swab for collection of specimens. The swab are to be plated without delay. In case of delay, modified Stewart's medium or charcoal agar should be used for transport of the swab. Medium employed is Bordet and Gengo or charcoal blood agar. Incorporation of diamine fluoride and penicillin into Bordet and Gengo medium makes it more selective. Plates are incubated in high humidity at 35 to 36 degrees centigrade for 3 to 5 days. Typical pulp like colonies of B. pertussis appear in 48 to 72 hours. These are confirmed by microscopy and slide agglutination with specific antisera. Immunofluorescence is useful in identifying the bacillus in smears from culture. The differentiating features of different species of Bordetella we studied. Now let's study about the serology. Rise in titer of antibodies may be demonstrated in paired sera samples by agglutination, complement fixation test, and immunofluorescence test. Antibodies in sera can be demonstrated in the third week. Detection of species IgA antibody in nasopharyngeal secretions by ELISA has been proposed in culture negative cases. Now let's study about the treatment. The organism is sensitive to various antibiotics such as tetracycline, uh, chloramphenicol, erythromycin and ampicillin. However, erythromycin is the drug of choice. Now let's study about prophylaxis. Immunization of infants and children with killed B. pertussis vaccine is very effective. Smooth phase 1 strain of B. pertussis is used for vaccine production. 3 intramuscular injections or intervals of 4 to 6 weeks are given before the age of 6 months, followed by a booster dose at the end of the first year of life. Vaccination is usually started at age of 6 weeks. Pertussis vaccine is generally administered in combination with diphtheria toxoid and tetanus toxoid. B. pertussis acts as an adjuvant for the toxoids, producing better antibody responses. Now let's study next is rarely neurological complications like encephalopathy and convulsions may occur. Children below 4 years of age who are contacts of the case should receive a booster dose along with a chromoprophylaxis of erythromycin for 5 days. Acellular vaccines containing PT, FHA, agglutinogen 1, 2, 3 of B. pertussis have been developed. They have fewer complications. The duration of protection with acellular vaccine is not known. Prevention by isolation of cases is seldom practicable as is the infectivity is the highest during early stage of the disease when clinical diagnosis is not easy. Next is let's study about Bordetella parapetosis. Pertussis. They are responsible for about 5% of whooping cough cases and generally produce a mild disease. Morphologically, they resemble B. pertussis but differ by their ability to grow on nutrient medium and pigment production. They are also antigenically distinct from B. pertussis. The pertussis vaccine does not protect against B. pertussis infection. Now, let's study about Bordetella bronchiseptica. It is motile with peritrichate flagella and has been found to cause 0.1% cases of whooping cough in humans. It can grow on nutrient agar and is antigenically related to B. pertussis and B. brucella abortus. Next we will study about Bordetella avium. It is the causative agent of coryza in turkeys. It can grow on nutrient agar. It produces HLT and TCT but does not produce ACT and PT. Finally finished. Any questions regarding this, you can ask me in the comment box. Subscribe my channel to know more updates. Thank you.